Welcome to the next lecture of our course on environmental biotechnology, and in this lecture number 45, we are going to discuss about the enhanced biological phosphorus removal process or EBPR. In this particular lecture, we will have an introduction to this EBPR process, that is the enhanced biological phosphorus removal, and we will also learn about how microorganisms they facilitate this enhanced phosphorus removal, and of course, we will also deal with the importance of this phosphorus removal etc. Now, enhanced biological phosphorus removal process or EBPR is considered to be one of the most modern wastewater treatment configurations, and in general biological wastewater treatment is considered to be the largest biotechnological industry in the world, and these biological waste treatment processes, these are essential for protecting human health and also the environment, and these waste treatment processes using diverse array of microorganisms are recognized as a viable source of energy, because through these waste treatment processes a large number of energy generation opportunities are provided. It also acts as a source of value added chemicals such as bioplastics and biofuel molecules, and also considered to be a source of non renewable resources. Now, EBPR, that is enhanced biological phosphorus removal, can only remove the contaminating carbon, nitrogen and phosphorus from wastewater, but it also recovers the phosphorus in the form in which it is more amenable to reuse than traditional chemical phosphorus precipitation used in the wastewater treatment technologies. And the process has become increasingly viable as a source of phosphate as prices continue rising due to the increasing demand and limited global reserves. So, on the one hand we have increasing demand for phosphorus, phosphate rather, and the declining natural resources, that is the rocks, which are considered to be the major source of phosphate in all the industrial processes. And also on the other hand, the new opportunities which are emerging because of the microbial activities that lead to not only the removal of the phosphates, but also provides us the opportunity to recover that phosphate and provide that phosphate as a kind of for industrial uses. Now, the increased what is the background of this phosphorus problem? Now, the increased input of phosphorus and nitrogen both from wastewater treatment plants and other sources may lead to disturbance in the ecological balance. The reason behind P and N, because P and N are released from diverse waste materials, and conventional waste treatment plants, unless they are categorically designed to handle the or to reduce the phosphate and the nitrogen contaminants, it would generally take care only about the carbons which are present there, like organic carbon mostly in the wastewater, and to some extent the nitrogen, but the phosphate which is there in the wastewater, unless it is the strategy or the wastewater treatment process is developed to handle the phosphates remain as phosphates and they are released into the environment wherever the wastewater treatment plants are discharging their the effluents. And when this phosphate containing effluents from the diverse wastewater treatment plants and other sources are released into the environment, they disturb the ecological balance, particularly in the lakes, which is found to be one of the lakes and the rivers are the most potent sink for this enhanced phosphate, not only for phosphate and also for the nitrogen pollutants, and slow flowing rivers, particularly water reservoirs and shallow regions of the marine waters along the sea coasts. So, in all these places which actually act as potential sink for the phosphates which are released by wastewater treatment plants if they are not dealing with or they are not treating the phosphate waste, and also other sources which are producing phosphates and they are releasing these phosphates. So, now this overly rich provision of inorganic nutrient like phosphate particularly, and also the nitrogen compounds like nitrate, and we will discuss about nitrate little later. So, that leads to a kind of an environmental problem which is called eutrophication. Now, eutrophication is considered to be a global problem. When you have enhanced amount of inorganic nutrients like phosphate and nitrates for example, in your water body, and if the water body is already, generally the water bodies are oligotrophic we call them, because they maintain a low organic carbon content unless it is polluted. So, the healthy water bodies which are considered to be oligotrophic or generally low in organic nutrients and also the inorganic nutrients like the phosphate and nitrate, a bloom of cyanobacteria and eukaryotic algae occur, and this bloom of cyanobacteria and algae occurs as a part or the consequence of the breakdown of the community homeostasis from the continued pollution of the oligotrophic water body. So, if you have oligotrophic water body for instance, and if there is a, for example, if we just consider that this is the water body. So, and if in this water body we have addition of phosphate and nitrogen nitrate, then the enhanced phosphate and nitrate. So, the enhanced P and N levels, that is the inorganic nutrient levels, what they will do? They will encourage the activities of cyanobacteria, and followed by cyanobacteria the algae will come, and this enhanced growth of cyanobacteria. So, cyanobacteria and the algae will be promoted. So, the enhanced growth of cyanobacteria and algae will actually disturb the ecological balance, that is called the homeostasis within this lake ecosystem. And essentially all this algae, when initiated by cyanobacteria, then algae, then the algal cells will die, and eventually the all biomass will be depositing, as I said, part of the sediment, leading to the enhanced concentration of organic carbon and nutrients into the lake, and gradually the quality of the water will deteriorate in terms of multiple parameters. The algae and cyanobacteria may produce toxins, they will add the organic biomass, and the availability of the organic biomass will lead to the decrease in the dissolved oxygen. So, the water body will become slowly anaerobic, and as you can understand that if the water body becomes anaerobic, then the life, the aquatic life will be all under threat, and eventually the lake will continue to be converted into a kind of a wetland, where in a couple of years, if we allow this to happen, then gradually all the lake water body will be filled with biomass, and eventually we will see that the grasses and all the plants, higher plants are growing on that, and eventually the lake will be converted to a wetland. So, that is the kind of consequence that we want to deal with. So, basically we need to handle this situation. So, eutrophication is a global problem. Now, here you can see that one of the eutrophicated lakes of Madurai, which was the picture was published. So, this is the early stage where you can see lot of the green algae is growing on them, and already the river is seems to be very polluted, and this pollution is adding nutrients further, and this algae, as I was explaining, will be allowing the deposition of the algal biomass, and eventually the lake homeostasis will be totally altered. Now, the well known consequences of the excessive nutrients, particularly the phosphate and nitrates, is the formation of the foam and slime, which disturbs the quality of the water bodies, the sea beaches, and the formation of toxin by the cyanobacteria. So, the water bodies are not suitable for public use. The oxygen respiration of the algae, if you have lots of phosphates and nitrates, then as I mentioned, the cyanobacteria followed by algae will start growing on them. Now, the algae will consume the oxygen, particularly during the night time, and the biomass decomposition by the bacteria primarily cause oxygen depletion in the water, and that will eventually lead to production of H2S, and because the residual sulfates will be all reduced as you have more biomass down there. the biomass will provide the organic resources, organic carbon and electrons, and the sulfate reducing bacteria along with other fermenters will lead to the sulfate reduction and production of H2S and creating lot of nuisance into that, because H2S will precipitate further metals and will stink and everything will be very much deteriorated condition. Now, oxygen shortage also causes a reduction of the nitrate to toxic nitrite, because as you have lower level of oxygen, because your algae is consuming oxygen, nitrates are going to be reduced to nitrite, and the nitrite is toxic, and the release of phosphate from the sediment will take place further. Now, processing of drinking water from the eutrophicated water bodies will require additional cost and efforts, because in many cases the natural water bodies are used for drinking water resources. So, if we pollute with respect to the nutrients like phosphate and nitrate, phosphate is more is a point of focus for today's lecture. If we pollute our lakes and water bodies which are used for drinking water purpose, then possibly we need to spend more money and more facilities to treat those waters to make them of drinking water quality, because then in that water perhaps more microorganisms have grown, because it is not oligotrophic lake anymore or oligotrophic river no more, because it is now rich in organic matter and lot of other activities are going on. Now, the crucial importance of phosphates in the fresh water. So, in natural water, inorganic nitrogen, for example, nitrate and ammonium and organic carbon are not obligatory for the growth of cyanobacteria. Why? Because under natural conditions, cyanobacteria they are capable of fixing molecular nitrogen, and they can also fix organic carbon, they can fix the inorganic carbon, carbon dioxide, and convert it to organic carbon. Now, concentration of phosphate is found to be a crucial importance, because that is what in generally is missing in oligotrophic lakes. Most importantly, the moment you provide or we provide phosphates into these ecosystems, water bodies, the gap is filled. So, the cyanobacteria were just waiting for the phosphates. So, the moment we have a continuous supply of phosphate in the lake or the water bodies, this will allow the cyanobacteria to start growing, because they are waiting for phosphate only, because they can otherwise manage with nitrogen and carbon sources. Now, eutrophication can only be prevented as at a concentration of phosphate less than 10 microgram per liter. Now, when we try to develop a system, because human and the environmental engineers particularly, they have worked for a long period of time. So, like more than 50 years or 70 years now, we see that the wastewater treatment technologies are developing, including the phosphate removal processes using microbes. And eventually, we have seen, now we have learned that it is not, as I mentioned earlier, not only removal of the phosphate, but we can actually recover the phosphate also. Now, reducing the global P reserves, the phosphate reserves, loss of P as human waste and other waste. So, basically 15 to 20% of world demand for phosphate rocks could be satisfied by recovering the P from these waste streams. So, all the wastes which are coming from the domestic areas as well as the industrial areas often contain high amount of phosphate. So, if we are able to recover that phosphate, it will be replacing possibly 15 to 20% of the rock phosphate. So, you can now imagine that actually replacing 15 to 20% of the world demand of the rock phosphates would actually mean that lot of mining activities, lot of other expenditures can be minimized. Now, what are the sources of phosphates or phosphorus in the wastewater? These are basically the fecal materials, industrial and commercial sources, synthetic detergents and other cleaning products, and also there are point sources like different wastewater treatment plants. Now, we will move into the P removal through the wastewater treatment plants. So, as I mentioned, over the past 50 years or so, many wastewater treatment plants have been designed and built around the world to reduce not just the organic carbon and nitrogen level, but also the phosphorus level using microbially based methods. Although the carbon removal methods are often well discussed, but the phosphorus removal methods remain missing. So, in today's class we are going to talk in detail about that. Now, what is microorganism mediated enhanced phosphorus removal? So, conventional wastewater treatment, which is basically up to having 7 to 12 milligram phosphorus per liter can remove 30 to 40% of the phosphorus biologically, and this phosphate is converted into the sludge, and the sludge can be removed and can be utilized for as a source of phosphorus. But we need to remove over 90% of the phosphorus. Removing 30-40% with normal conventional wastewater treatment plant, where basically it is carbon removal, and during the carbon removal also the organisms they accumulate some phosphorus and they precipitate as a biomass sludge. So, we need to bring down the phosphate concentration with the permissible limit to 0.5 to 1 milligram phosphorus per liter, and that sounds to that goes to around like 90% of the P present in the wastewater treatment. Now, this can only be achieved by two methods. One is the chemical precipitation method as the phosphate salt, and of course, we are not right now focusing on that. The other alternative is that the incorporation of the phosphorus or phosphate into the biomass of the sewage sludge, because compared to nitrogen compound which can be biologically catalyzed to produce nitrogen, and nitrogen can leave the system, phosphorus or phosphates cannot leave the system. So, it will be deposited within the biomass. So, ideally if we look at the wastewater treatment process, so this is for example, the wastewater where we will allow the specific microorganisms to grow, and once these microorganisms are able to encounter the phosphates which are present over there. So, they will be able to accumulate the phosphate as polyphosphate body. So, these materials are called polyphosphate body. It is a long chain of polyphosphate groups, like for example, if you see the ATP molecules where we have only 3 phosphate groups, similar type of anhydride bonds are there, but there may be 40, 50, 60, 80, 100 phosphate molecules, I mean more than that are tied together, and these are large cellular deposits which we can observe under the microscope, that the cells which are expert in converting and accumulating phosphorus, phosphate and converting them into polyphosphate bodies can be seen under the microscope very easily with appropriate strain. So, now if we allow this bacteria to grow with phosphate, now this water might be having high P. So, we will see after some time that all these bacteria they will deposit this long chain of polyphosphate bodies within them, and they will grow. These bacteria will grow more. So, as they grow more in number, so we have more number of cells appearing, and once you have more number of cells appearing and growing, so they will accumulate more phosphates, and what will happen, the water that will eventually come out of the system will have the phosphate level which is close to the desirable limit, that is 0.5 to 1 unit of per milligram of per liter of water. Now, this removal is achieved by encouraging this type of bacteria which I have shown here, and this type of bacteria they are called actually phosphate accumulating organisms or phosphate producing bacteria. So, they accumulate the polyphosphate bodies. So, they take the phosphate, the soluble phosphate, they transport it inside the cell, and then they produce the polyphosphate body. So, they have the appropriate metabolic mechanisms to produce. This is also a very kind of an exclusive property, that all bacteria present in the wastewater will not have this property of producing excessive polyphosphates, which will actually meet the industrial wastewater treatment level. Now, the EBPR or EBPR, the enhanced biological phosphorus removal process, uses the capacity of these bacteria which are known as the polyphosphate accumulating organisms, or conventionally also known as the PAOs. So, these PAOs are the organisms who are responsible for accumulating the phosphates as polyphosphate bodies. Okay. Now, along with the polyphosphate accumulating organisms or polyphosphate accumulating bacteria, they mainly belong to bacteria. So, we conventionally call them as polyphosphate accumulating organisms. There is another interesting group of organisms which are very closely associated with the growth and activities of the PAOs. These are called glycogen accumulating organisms. GAOs. So, both these groups of organisms are selected for during the wastewater treatment processes which are developed for the removal of the phosphate. So, now if we look at the wastewater treatment processes which have been developed for phosphate removal, or we say enhanced phosphate removal. Why it is enhanced? Because normal wastewater treatment can remove 30 to 40% of the dissolved phosphate, but we need 90% of the 90% removal of the phosphate. So, ideally, as you can see over here, it is written that the anaerobic aerobic cycle is there. So, during the anaerobic aerobic cycling is there, that means, so your wastewater, so the wastewater which contains the extra or enhanced level of phosphate and we want to remove it, these wastewater must pass through a set of tanks or reactors. So, these will pass through, the wastewater will pass through the set of tanks, and individually these tanks will have the following designations like anaerobic, anoxic, aerobic, and then again some sludge will come back here and some water will flow out. So, the inoculum kind of thing will come back and will be added to here and the water will flow through. So, in between the water will be transferred. Now, the interesting point is there that while under aerobic condition the phosphates will be removed by the PAO, this glycogen accumulation will occur during the anaerobic process. Now, why do we need this glycogen accumulating organism? Actually, we do not possibly targeted these glycogen accumulating organisms initially, because we were targeting the phosphate removing organisms, but we realized that during this process of cycling of the water, because that only encourages these PAOs to grow and act properly, they allow these glycogen accumulating organisms also. Actually, these PAOs, they have a very intricate and interesting relationship with GAOs. Why? Because PAOs will take up the phosphate and will produce the poly P under the aerobic condition, but we need to have certain kind of cycling in order to facilitate the enhanced phosphate removal. So, the GAOs will actually partly they are more active during the anaerobic process. So, during that time they will produce the lipids. So, the glycogen accumulating bacteria, they will produce the glycogen deposits, which will be then utilized as a source of energy or source of the carbon and electrons for other bacteria like PAO as well. So, we will discuss about that. This intricate relationship between the anaerobic and aerobic conditions are essential for the natural enrichment and performance of these bacteria, which are called basically the phosphate removing organisms or phosphate accumulating organisms. So, the best performance of these organisms will be achieved only when we have the wastewater treatment system, which actually cycles between these aerobic and anaerobic systems. Now, the EBPR takes advantage of the complex microbial ecosystem to facilitate this process. Now, what is this complex microbial process? And this complex microbial process was initially not known. Initially, scientists thought that it might be by some designated group of bacteria or other members, fungi, etc. They are playing role in phosphate removal. But as soon as the modern molecular methods, molecular microbial ecology methods from single cell microbiology to omics approach, that has come in a big way to understand the complexity of the process. And many of the important organisms are identified and their main functions and metabolisms are also determined. So, what we found that we cannot assign any specific bacteria directly for the function of the phosphate accumulation in this case. So, there are some microorganisms. Now, the some microorganisms must have some identity. Now, these are generally called PAOs. These are the key organisms in this process and remove the most of the P from the wastewater, and they contain a high fraction of biomass P. So, they accumulate like up to 12% compared to 3% dry weight in non-PAO. So, the PAOs are the catalytically responsible organisms. Now, remember, all organisms are not PAO. There are some organisms which are PAOs. These organisms can accumulate up to 12% P dry weight basis compared to only 3% non-PAO organisms, and can be removed with surplus sludge for further processing and P recovery. Now, this 12% dry weight P is significant, and if we can remove, and indeed we do that. So, if we remove that sludge containing the bacteria which are loaded with phosphate, the phosphate can be removed successfully. Now, what are these organisms? This most well known group of PAOs are considered to be Candidatus Accumulibacter phosphatis. A Candidatus means the taxonomic description of this group is not yet complete, and they are found to be members of the class beta proteobacteria, and the major identifiable lowest taxonomic level identity is Accumulibacter. So, we can designate them like Accumulibacter, and the pure cultures of Accumulibacters are not yet available. They are mostly uncultivable organisms. And besides Accumulibacter, gram positive Tetrasphaeria related organisms and certain other Actinobacteria, Streptococcus like members are also there, and they play a very important role. However, this Tetrasphaeria is also considered to be, which is a member of Actinobacteria, another PAO. So, as a PAO we can identify the Accumulibacter and the Tetrasphaeria. Now, the Accumulibacter perform an unusual metabolic balancing act which gives them the selective advantage in the dynamic condition of EBPR. Now, if you normally grow Accumulibacter, perhaps it will not execute that function of up to 12% dry weight P accumulation, possibly it will not do that. It has to be cycled between two conditions, aerobic and anaerobic. So, let us first see under aerobic condition what happens. Under the aerobic condition, growth and uptake of and the storage of the phosphate, which is in the form of polyphosphate bodies, occurs in the subsequent condition where nitrate and oxygen are present. So, ideally using nitrate as electron acceptor and oxygen as electron acceptor, this aerobic mode of metabolism by this bacteria, this Accumulibacter type bacteria, they facilitate the accumulation of the P, uptake of the P and accumulation and its conversion into the polyphosphate body. So, ideally they are all aerobic bacteria, and they often executes the property of nitrate reductions also. So, here is a point that we can actually deal with the nitrate level also to some extent. Now, under anaerobic condition, now the same culture, same water will be transferred to the anaerobic stage. Now, under anaerobic condition, substrate uptake, that is the whatever organic carbons are there, that will be taken up, and the formation of polyhydroxyalkanoids or PHA, which is a potent bioplastic molecule, as a storage product will be produced without growth taking place under the anaerobic state. So, if we take the same culture under the anaerobic condition, we will see that the bacteria, they are actually removing the carbon from the water and converting these carbons into the polyhydroxyalkanoids, and during that time, this is anaerobic, they are possibly using the internal ATP generated because of the some of the polyphosphates bodies may be hydrolyzed, giving them some ATPs to perform these functions, where the small organic molecules will be taken up and they will be converted to the internal deposits of PHA. Now, interestingly, under this anaerobic condition, most other fast growing bacteria are inactive, because it is a wastewater coming. So, the wastewater is already mixed with this PAO containing inoculum. So, the PAO bacteria, they are very smart. So, they will survive, they will function not in terms of the P removal, but in terms of the formation of the PHA internal lipid within themselves. Now, these organisms or these culture, when will subsequently transfer to the aerobic condition, they will execute rapid growth under aerobic condition, and they will start accumulating phosphate. Now, the scientists they have studied the core microbiome, that what are these microorganisms, because it is a complex system. So, we have found that PAOs, while incorporating to P removal, they are important for P removal, constitute only a minor part of the EBPR community in full scale plants. So, across the several countries of Europe, we see there are multiple phosphate removal systems or wastewater treatment facilities which use this EBPR system. So, what we have found that in many cases this known Accumulibacter etc., they constitute only a small part, like 15 to 25% of the all bacteria present over there. So, what the other microbes, other bacteria etc., are doing there? The other microorganisms involved there is carrying out the important other functions like hydrolysis, fermentation, nitrification and denitrification, and other processes which are possibly important for the growth and activity of the PAOs. So, this is the data out of this review paper by Nelson in 2012, which shows that out of 25 wastewater plants from Denmark, they have reported that the core microbiome is there. So, you can actually look at the legend and then find out these are the genera which are given, and the functions carried out by them are provided here. We can see that many of them are fermentative, denitrifier, ammonia oxidizers, nitride oxidizers. So, there are many different functions, and all these different functions which are some of the functions are may not be directly related to phosphate removal, but when we look into the functioning of the entire system, we understand that these are also very essential. Now, the EBPR has emerged as a model system. Now, an in-depth knowledge of the microbial communities in the process and their interactions help to improve plant design, how these wastewater treatment plants will be designed, how they will be operated, and how we will be troubleshooting based on the variations of certain parameters, by enhancing our understanding of the critical aspects of the process. So, these processes are satisfactory removal of carbon, nitrogen, phosphorus, micropollutants and pathogens, good solid liquid separation, stable operation, and many fundamental questions regarding the microbial ecology of the communities are to be investigated. Now, the existence of a full scale EBPR core microbiome, which suggests that only 10 to 15% or maybe 12 to 20% is the Accumulibacter. So, other organisms are there. So, that a limited number of microbes are critical, but other microbes are important in order to control this process. Now, knowing these microbes, that is the selected organisms which are responsible for actual the P removal and others who are supporting them. So, knowing these organisms and their interactions, how they are interacting with themselves, enables the fundamental ecological insights, that can be transferred to other plants and perhaps even other similar microbial ecosystems. And hence, the key questions that we need to address for each of these core species in the major functional groups are delineated, and these include the transformation processes which they perform. What are the solid liquid separation properties like flocculation or biofilm formation? How can their presence be manipulated, that is removal, control of unwanted bacteria, etc. So, the model has been developed which suggests that if we have the inputs, we can have multiple outputs, including the species analysis, the community analysis, the system analysis, and then you have the outputs which are the purified water, and you have the different products and also the biomass. Now, we will see the life under the highly dynamic condition. It is highly dynamic because we are recirculating the water, wastewater through these anaerobic and aerobic systems. So, EBPR exploits rapidly changing environmental conditions. So, this is in the industrial scale setup where we actually transfer the water from anaerobic to aerobic and again aerobic to anaerobic, all these two systems are used. So, it exploits rapidly changing environmental conditions, and these changing conditions are required because that select for the diverse range of organisms. Now, again and again that point will be coming in mind, that why do we need to recycle between these aerobic and anaerobic conditions? It is required because that shifting the conditions will allow selection of diverse organisms, which will be required for the desirable efficiency of the PAOs. Now, ideally what we see that the system can be operating with the wastewater input moving into an anaerobic tank first, and then to an anoxic tank, and then again to the aerobic tank, and then from the aerobic tank the water will move into the clarifier, and from the clarifier the water will further be discharged, and from the aerobic tank some of the material will be recirculated, and some sludge will come from the clarifier and it will consider as a return sludge, and the return sludge will come to the and mix with the wastewater as possibly as an inoculum, and the left part other part of the return sludge will be used as a kind of the product from where the phosphate can be removed. Now, if we see the processes which are going on there will be processes like anaerobic, anoxic, oxic. Then again as I mentioned that this part will be circulating at least 4 times. So, these are optimized situations. So, what Nielsen in their paper has actually mentioned that these are kind of a model situations, model conditions which are being practiced across several wastewater treatment plants. Now, down here we have the several organisms. Now, these organisms are the most prominent organisms and they are relative abundance, like the color code here saying that no growth, like the white color means no growth, light green is low growth rate, green is higher growth rate, and the shaded green is active, but without growth. Now, the activity level plays a key role for establishment of the selective pressure, controlling the community composition and also function. As you can see, many organisms including nitrifier, denitrifier, filamentous hydrolyzing organisms who can produce exozymes, and exozymes are involved in hydrolyzing different complex polymers, and then you have the PAOs of 2 phylogenetic clades, Acinetobacter clade 1 and clade 2. So, they are dark green in many, and most of the cases they are dark green in oxic conditions, but they are also active, but they are not growing. For example, Acinetobacter, if we see Acinetobacter is active, but they are not growing here. Okay. Similarly, if you see Streptococcus very active in anaerobic condition and growing also, but it is growing with a low growth rate, and same thing is true for Thoira and microthrix as well. So, ideally the most microorganisms in EPBR configuration found to be versatile and potentially active in more than one phases, and that is possibly one of the major characteristics of this entire process. Now, this entire process can be defined as a game of 4 major players. Who are the 4 players? Number 1 is the denitrifiers, which are the facultative anaerobes, most active under anoxic condition, as the organic substrate concentration is high than the aerobic tank. So, they are very active under the anoxic condition, because the redox potential under anoxic condition possibly favors them, and they utilizes the organic compound, or they utilize the organic compound and they reduce the nitrate. Microthrix active in all tanks. So, aerobic, anaerobic, anoxic, everywhere microthrix are there. They take the substrate, use it under anaerobic condition, reduce nitrate to nitrite in the anoxic tank, grow aerobically in the oxic tank, either or store material or hydrolyze the products, produce the exozymes in all tanks. So, this exozyme production is very critical, because this is essential for degrading or hydrolyzing all the large polymers of the proteins and carbohydrates and lipids, etc., which are then making their use in the subsequent steps. So, Streptococcus is a versatile fermenter, most active under the anaerobic condition, but also able to grow under aerobic condition. These ferments produces different metabolites, and possibly these metabolites could be of use by other organisms including the Acinetobacter also. Now, Acinetobacter, which is most active and able to grow profusely with phosphate accumulation under oxic condition, but it can remain active, but they do not grow under anaerobic condition. So, under anaerobic condition it releases some amount of orthophosphate, okay, and take up substrate which are present like the volatile fatty acids or small acetate like compounds, and store them at the polyhydroxy alkanoids or lipid bodies. So, under anaerobic condition Acinetobacter plays the role of GAOs like glycogen or PHA accumulating organisms. Under anoxic or aerobic condition, Acinetobacter take up orthophosphate and utilize the stored energy for the growth. So, this is the beauty of these 2 phylogenetic groups of Acinetobacter, clade 1 and clade 2, that one of them is very active during anoxic condition, another is very active during the purely oxic or aerobic condition. So, that means even under complete aerated condition and also in partially aerated or low oxygen concentration level, the different Accumulobacter strains are capable of removing the phosphate from the system. So, for this lecture, these two articles will be very useful. And finally, in conclusion, the developments in technologies to study the microbial communities that have taken place in the past 2 decades have revolutionized the way we analyze, understand and manipulate microbial communities, and we have learned some aspects and a basic concept of that. The presence of the core population in EBPR plants suggests that the data obtained from detailed studies of the limited number of systems is actually transferable to the field as a whole. So, this is a very interesting outcome that we have learned from EBPR system, that other microbial systems or microbe based wastewater treatment and environmental systems can be developed using a similar concept. This could just be the start of a we call systems microbiology, that instead of finding one species responsible for one function. So, here multiple species, multiple functions, but helping into towards achieving the broader goal, that is remove phosphate and allow us to recover the phosphate. And finally, integration of these genomics, metabolomics and other systems level data leads to the better understanding of the communities with more predictive power of for the benefit of the wastewater treatment plant, and eventually produce tools for optimizing and controlling the processes, carry out troubleshootings and develop new process designing. Thank you.